Okay, welcome everyone. It's my great pleasure to open this year's first Provost lecture, and which will be given by Professor Keiko Kono. And so this is a very special occasion, so we're here to celebrate Keiko to be promoted to tenured associate professor. Congratulations. So I will hand this over to Tomomi, who is going to chair this session. But before I let Tomomi take over, I just want to quickly introduce, many of you already know, so the Provost Lecture Series was started in October 2022. And so this is specifically designed for OIST faculty members. And so we use this to celebrate OIST faculty members' different milestones, and for those who got promoted to associate or full professors, and for those who have received special awards, and also for those who are about to retire. And so it's kind of, you can see, so since November 2022, and today's lecture will be number 21st. And so the last one was given by Professor Shintake on his EUV lithography last December. And so before I forget, so this year we have, so we're planning for many Provost lectures in this year because we have many professors got promoted in the past few months. And so the next one will be given by Tom, who is promoted to full professor. That will be February 12th. And so just one more slide. I want to also take this opportunity to thank many people who made this Provost Lecture Series possible. So especially, I want to thank everyone from the Office of the Provost, especially Masukawa-san, who has been coordinating and making sure everything is coordinated on time and properly. And also starting this year, everyone in the Provost office is going to participate to support this event. And also so many people from CPR. So for example, Michael Cooper, Jeff, and also Scotty, I got this new picture. I replaced the old one, and Rhett and Kari for designing the poster. And also people from the core facilities, so especially Patrick and his team, so they have been helping us to 3D print and design the plaque for each speaker. So without further ado, Tomomi.
My name is Tomomi Kiyomitsu. Today I serve as a chair. So first, Keiko, congratulations on your promotion to the tenured associate professor. It's a great pleasure and honor to serve as a chair for your provost lecture. And as you might know, every day we look at Keiko's picture when we walk through the tunnel gallery. But I'm not sure whether you know Keiko very well or not. That's why let me please introduce Keiko's career and research briefly. Keiko was born in Okinawa Prefecture, Japan. And then she spent 1 year in US with her family. Especially I like this picture because this nicely suggests she was a very curious child. Then after this 1 year stay, the Keiko's family returned to Japan, and Keiko grew up in Japan. And when she was young, she enjoyed a lot of different activities, including ballet. I like these pictures. Yeah? Yeah? Then now I understand why Keiko always had a good posture. I have a bad posture like that. But Keiko, always something like that, right? Yeah, yeah. Now I understand. Okay. Then she entered the University of Tokyo, then got a PhD in 2005. Then she joined David Perlman's lab at Dana-Farber Cancer Institute in Harvard Medical School, where she studied her research related to membrane damage response. She also enjoyed a lot of activity in Boston, including this informal scientific meeting for Japanese postdoc in Boston area. Fortunately, I met Keiko. However, in those days, I did not expect we will have a laboratory in OIST. Also, I will serve as a chair for you. So life is very unpredictable. Then Keiko got a position, lecturer position in Nagoya City University. Then Keiko married Yosuke. Where is Yosuke? Is he here? Very gorgeous. Yeah. And during this period, they got 2 kids. Then Keiko and her family joined OIST in 2017. This is current picture. Now kids grow very fast. And they are currently studying in Onna Elementary School together with many children of oysters, including my daughter. As you might know, Moriyama-san, Yosuke is the current president of PTA in Onna Elementary School. And Moriyama-san has spent a lot of effort and time to support OIST parents and children. So I really appreciate your contribution. Thank you very much. And these are Keiko's achievements during this period. So Keiko published 14 papers, including this famous Nature Aging paper. And she graduated 6 students as a primary supervisor and 2 students as co-supervisor. This is amazing. And she has also contributed to OIST as a member of faculty council and other committee members. So we really appreciate Keiko's outstanding contribution. So this is my last slide. So today's title is Cell Membrane Damage and Aging. As you know, cell is a minimal functional unit of all living organisms, which contains DNA and proteins and endomembrane system inside. But cell must be wrapped or closed with a lipid bilayer called cell membrane or plasma membrane to distinguish itself from others. Keiko and colleagues recently found cell membrane damage are related to cellular and organismal aging. Keiko's long-term scientific goal is to reveal the link between cancer senescence and plasma membrane. I stop my talk, and yes, please welcome Keiko with applause.
Thank you, Tomomi, for the great, maybe a bit embarrassing introduction. I really appreciate it. And thank you all for coming here despite your busy schedule. I really appreciate it. Thank you. So, and Amy, thank you very much for giving me a great opportunity to present my work and for everyone who kindly contributed to this event. So today I'm so excited because, well, actually 9 years ago, I was exactly interviewing in January here at OIST to apply for the faculty position. And actually, indeed, I was applying here 10 years ago again. So I applied here twice. The first year I got interviewed, but I didn't get on the faculty offer. But still, I applied here a year after again because I fell in love with OIST culture. So when I say OIST culture, I mean OIST people. And after like 8 years, I got to know many OIST people, not just researchers, admin side. I got to know many people, and I love OIST more than I did 9 years ago. So it's truly my honor and the pleasure to get tenured here, to be allowed to stay here until the rest of my career. So today, I'm so excited to share the very first day Kono unit lab photo with you guys. So yeah, only Yosuke and myself and family, and Hitomi, RUA Hitomi was there. And after 8 years, the lab has grown. And also my kids were grown as well. And today, I would like to, my current central question is, how do we age and how do our cells age? So let me explain how I get to this question. Because 2 years ago, I got the privilege to be featured on the cover of the image. And the central finding here was, so here, we took the motif of Japanese traditional art of gold stitching, kintsugi. So here you see the pottery, and here you see the cell. And the cell membrane is damaged, and damaged membrane itself can be stitched together, but the cell will not go back to the original young status, but it irreversibly changes its quality to aging. So that is the central message of this paper. And I got the cover image of the sister journal. It sounds really fancy, but actually, I would like to share this experience. Starting from the initial submission, it took more than 10 years to get accepted. And this paper was rejected by 8 journals, meaning that, back in those days, when I was younger, I was thinking life is like this, either failure or success. But in reality, after experiencing applying OIST twice and also submitting the paper, keep submitting for more than 10 years, I realized the real life is like this. Just failure, 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 keep going, going, going. And at some point, I got some success. So today, I'm so excited to share my story about full of failure and unexpected twist in my scientific career. All right. So in the plasma membrane damage research field, actually it has like 30 years of history. And plasma membrane damage is known to induce 2 cell fate, either repair and recovery or the death. But we found there is the third cellular outcome, which is cellular aging, cellular senescence. So now, we get to this conclusion, but initially I wasn't working on aging or senescence at all. Actually, I was simply working on how do our cells repair the damaged cell membrane. So I was interested in the membrane repair process when I got a position here 8 years ago. So our body is composed of so many different cells, so many different cell types, so many different cell shapes, cell function, but all the cells are surrounded by the very thin plasma membrane. So as you see here, so it's a cultured human cancer cell. So the red signal indicates the chromosome, DNA, so genetic information, and it's surrounded by the green, which is the plasma membrane. So plasma membrane is fundamental to life and cell because plasma membrane is a border of the environment and cell life. But actually, it's very, very thin. It's only like 5-10 nanometers. So like you see very thin black line here. So this is a cultured normal human fibroblast cell, and it's an EM image, electron microscope image. And this thin layer is the plasma membrane. So it's actually thinner than the soap bubble. Therefore, it can be very easily damaged. And therefore, in our regular activities, in a physiological context, our cells are quite frequently damaged, and it has to be repaired. And the best known context is a muscle contraction. So if you do like running for your health maybe, or the muscle training, your muscle cell get tiny, teeny nanoscale damage on your cell membrane. And usually that is immediately resealed within seconds. So by doing that, you can maintain your body homeostasis. But not just muscle, like many other processes like heart beating, breathing, blood streaming. So any mechanical stress context, you can get a tiny, teeny wound on your plasma membrane. So let me explain like how important the membrane repair process is. So here you are looking at the cultured normal human cells, and that is treated with the detergent, extremely low concentration of detergent. So you can imagine like 1,000 times diluted shampoo. Even if you shake it, you will not see any bubble. So it's almost like a water. That diluted detergent, and then usually the cell can survive in the presence of those detergent. However, if you decrease the medium concentration of calcium ion, that actually calcium is required for the plasma membrane repair process. So even mildly decrease the calcium concentration, if you add the diluted detergent, your cell will just immediately pop. Therefore, we always have calcium around our cells, so we are not aware of it. But actually, plasma membrane damage repair response is fundamental to our life. And if this mechanism is fully impaired like this, your cell will just immediately pop and die. And if this mechanism is only mildly impaired, you will have diseases like muscular dystrophy. So there are several proposed models for the plasma membrane repair. So for example, actually depending on the context, what kind of damage, what kind of cell repair mechanism is different. But the two major mechanisms are evolutionarily conserved. The first one, calcium ion dependent vesicle fusion. So if you remember the cell biology textbook, so maybe you all know that the calcium ion concentration in the cytosol, cell inside of the cell, which is very low. And calcium ion concentration is 10,000 times higher in the environment. Therefore, the very first thing that happens after the membrane rupture is the high concentration of calcium getting into the cell. And triggered by that, vesicle fusion happens, and the damage can be patched. This is the one major mechanism of membrane repair. And another mechanism is the protein called escort filamentous protein that forms like a coil-like structure at the neck of the damaged membrane. And escort can pinches off the damaged membrane. So these calcium ion and escort protein are the evolutionarily conserved plasma membrane repair responses. And like 10, 15 years ago, when I was in David Pellman's lab, so there are some people in the field working on the membrane repair mechanisms. So mainly they are working on the oocyte, like the egg of like some fish or sea urchin or frog. They are relatively like a big cell. They put the cell under the microscope and make a hole with the glass needle and observe how they are resealed. Therefore, they needed to look at the protein one by one, which protein is involved in the repair process. Therefore, the progress of the field was very slow. So I wanted to utilize completely different system, which is OIST. So why OIST, you might ask, right? So if you are interested in the human, ultimately, you should work on the human cells. Why you work on the OIST? Well, OIST is amazing, right? So OIST makes all amazing products. Definitely, we do love OIST, but not just that. Actually, OIST is a powerful tool to identify the gene set required for the completely newly identified and evolutionarily conserved cellular processes. So these are all Nobel laureate. And the people highlighted in blue utilized OIST to identify the gene set required for the fundamental cellular processes highlighted in red. Therefore, if the process is evolutionarily conserved and fundamental, and yeast is definitely one of the ideal model organisms to identify the genes involved in that process. So if you are not the biologist, you might wonder, like, why do we use the model organism? So that is because it's simple. So you can imagine model organisms are like a bike, and human cells are like a car. And if you want to investigate the function of the gene tire, for example, molecular biologists always knock out the genes, right? So take out one tire, right? And see what will happen to the bike. So then definitely they stops going, just fall down. Then they stops. So it's very easy. We can conclude that, yes, the gene tire is required to go forward. But in case of the more complicated higher human cells, we have 4 tires, meaning that a more overlapping genes in the complex cells. Therefore, if we take out one gene tire, we still have 3. So the car will not stop. Well, it can be slowed down, but it can still wiggle and move. Therefore, we cannot conclude that function of the gene tire. That's why we use the model organism, and the simplest could be the yeast. So back then, 15 years ago, people were using oocytes and a glass needle to make a hole and study membrane repair process. So I wanted to use yeast. But yeast is like only 5 microns, and it has a very thick cell wall. So it's almost impossible to use a glass needle. So then what kind of method can we use? So actually, I'm the very first person who decided to use yeast in this research field. Therefore, we needed to establish everything when I was in David Pellman's lab as a postdoc. And I developed 2 assays. So one is a laser damage assay. So we put the living yeast cell under the microscope and hit it with a very strong laser and create the sub-micron scale damage and observe how they are repaired. And another assay is, again, the extremely diluted, 1,000 times diluted shampoo type of detergent. So this concentration of detergent is the lower than the micelle concentration. Therefore, it will not directly induce the damage, but it will accelerate the induction of natural damage. So by doing that, I can introduce the local plasma membrane and cell wall damage. So combining these 2 assays, I try to identify the gene set required for the plasma membrane repair. So to do that, I used the yeast gene deletion library. So yeast has 6,000 genes, and there is a library in which each single genes are knocked out, and the cells are just put in each well and frozen. So we can easily purchase it. So I got it and played it onto the regular plate or the plate with the membrane damaging reagents. And if the genes knocked out in the cell are required for the survival after membrane damage, so those gene deletion mutant cannot grow only in the presence of membrane damaging plate. Okay? In this setting, I identified 48 out of 6,000 genes as the genes required for the survival after membrane damage. Like here you see the gene deletion mutant, and they cannot grow only in the presence of membrane damaging reagents. And so among them, I thought at least some of them could be the membrane repair factor. So I was checking the format paper or database whether what kind of functions are assigned for each gene. And there was a surprise here. So plasma membrane damage response seems to be associated with completely different cellular process, which is cell aging. I actually did the database search and realized that 1/3 of my screening hit are previously reported as the aging regulator. So I wanted to identify the membrane repair factor, ended up identifying many aging genes. So how can we explain this? So here, let me remind you what was the cell aging, so senescent cell. So here you're looking at the cultured normal human fibroblast, young cell and the senescent cell. So actually this is one cell, and so this is one cell. So size and shapes are completely different. And normal human fibroblast usually can divide about 50 times, and it stops growing forever. So in contrast, cancer cell can divide forever, right? But the normal cell has a limit in its division. So that's the primary definition of the senescent cell, irreversible cell division arrest. But if you hear like senescent cells are a non-dividing cell, you might think it might not be so bad, right? If the cells are just sitting there, it doesn't sound so bad. But actually they're bad because they produce a lot of senescence-associated secretory phenotypes. So it's cytokines or chemokine or growth factors. So it's something like a hormone. By definition, it's not a hormone, but the soluble protein, and that deliver the message to the surrounding cells. And the primary function of those proteins are the upregulation of the immune response. And if that immune response is upregulated at the right level, that is beneficial for the body. For example, like a tissue wound repair can be accelerated by senescent cell and also SASP. But the bad function of senescent cell is like the acceleration of age-related diseases, including cancer. So like 10, 15 years ago, multiple groups reported in an animal study that the removal of senescent cell, either by the genetic recombination or by the drug treatment, can rejuvenate the several body functions. And based on that, some people hypothesized that maybe like same thing can happen in human cell. So this is still a frontier. There are some evidence supporting this conclusion and others contradictory to it. So I think at this moment it's still a frontier of the research and a controversial thing, but potentially removal of senescent cell can attenuate various age-related pathologies. But you might think, but actually the rejuvenation sounds like a science fiction. So can it really be possible? I think it might be possible because actually dogs age, cats age, we human age, so we tend to think like every organism age. But it's not really the case. There are some organisms that are ageless. So one example is like the naked mole rat. Do you guys know this? So they are rodent, African rodent. They live underground in Africa, and they live very long. Like normal rodent live only 2-3 years, but they live 30-40 years. And their mortality, so their death rate does not increase by age. It's always flat. And no cancer formation, and they can deliver the kids by the end of their life. Well, actually, they don't look so young, right? Rather old, but actually, they can maintain their youth during the entire life. Therefore, actually, life and aging can be the separable phenomena. So it's possible if we learn the trick from those organisms, maybe we can slow down our senescence aging processes. All right, so let me remind you what is the best studied senescence mechanism. Maybe you've heard about the telomere shortening thing. It also got the Nobel Prize, so I believe almost everyone knows this. So every time the DNA is replicated during the process of cell division, the end of the DNA is not fully replicated. Therefore, every time you undergo cell division, the end of the telomere, the end of the DNA, gets shorter and shorter and shorter. And at some point, the shortened telomeres are detected as a damaged DNA. And how does the short telomere induce cellular senescence? So the DNA damage repair pathway is activated. So if you are the cell or molecular biologist, you can easily understand what this means. But if you are not the expert, you might think, what the pathway? What is activated? So actually, cells are the system. And you can imagine something like a domino. So here you have a protein, surface protein, and some protein, like a hormone or SASP, can bind to the receptor, and they can bind to the target protein, another protein, and changes its structure, and it will further activate other targets, protein, and then domino kind of thing happens, and ultimately that will change the gene expression profile, and the cell changes its behavior. So this is what happens in a cellular signal transduction in the cell. So if the telomere, when the telomere is long, it is usually covered by the protein. Therefore, the DNA damage sensor protein cannot be accessed to it. But when it's shorter, the exposed DNA can be bound and activated and sensed by the sensor protein. So then this protein further binds to activate downstream, 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 and ultimately changes its gene expression profile to cellular senescence, cell aging. So this is how we see that telomere shortening induces cellular senescence. Okay? So, and you might think, like, if cellular senescence induces aging, we should simply inhibit cellular senescence. That should be beneficial for our health. You might think that way. But actually, that's not the case. If the cells do not undergo senescence with short telomeres, what will happen? Actually, the shortened telomere can be stitched together, and you will get abnormal chromosome structure. And if this cell keeps dividing, you will easily get cancer. Therefore, those abnormal chromosome structures the cell with the abnormal chromosome structures should undergo cellular senescence and stop dividing. Okay? Therefore, cellular senescence is actually a defense mechanism against cancer as well. And actually, there are many triggers of cellular senescence. So you know, like sunlight irradiation, smoking, inflammation, reactive oxygen species. So many stress will ultimately activate DNA damage repair pathway. And then cell undergoes senescence. So actually, that was the common sense back then. All right, so let me get back to my own work. We found, I tried to identify the membrane repair factor, ended up identifying the many aging genes in my screening. So what's the link between plasma membrane and cellular senescence? And back then, DNA damage was a very well-established cause for the cellular senescence. So we thought maybe not just DNA. Membrane damage can also induce cell aging. And we tested that. And the answer was actually yes. So here you're looking at the x-axis as a number of cell division, y-axis as a cell viability. And black line indicates the wild-type cell. And east wild-type cell stops dividing after like 25 or 30 times of cell division. Cells start dying. And my screening hit, that is weak to the membrane damage, had a shorter cellular lifespan, age early, even without membrane damage. And if we upregulate those factors, we can extend cellular lifespan for about 17 percentile. And in the presence or absence of membrane damage, we see the same tendency. So these results suggest that detergent-dependent membrane damage can induce early cell aging. So, but it's just detergent, right? So I wanted to test different types of mechanical stress and membrane damage. So we took the glass needle And actually, really talented PhD student, Kojiro Suda, took a single yeast cell with a glass needle and squeezed it on a glass cover glass 3 times, and look at the consequence of those squeezing stress. And what he found is this type of squeezing mechanical stress can shorten the cellular lifespan, so they can age early. So altogether, the mechanical stress or membrane damage can induce short cellular lifespan, so early aging in yeast. So next, we wanted to test whether that can also be the case in a human cultured cells. And answer was actually yes. So here, we see the human normal fibroblast cultured in the lab and treated it with 1 day with extremely diluted detergent, like 1,000 times diluted shampoo. Cells are all viable. They are not dead, and wash it away, put it back to the normal growth condition, and waited for 16 days to see the cell undergo senescence. And we do see like young cell grow well, but the senescence plasma membrane damaged cell stops growing, and we see the senescence marker. This blue color is increased. And also another hallmark of senescent cell, like arrest of the DNA replication, was observed. And also we see the senescence regulator protein upregulation, P53, P21, P16. And also we see the upregulation of SASP, so senescence hallmark, inflammatory cytokines, chemokines. So these results suggest that the detergent-dependent plasma membrane damage can induce cellular senescence. And not just the STS-dependent senescence, we also look at other types of plasma membrane damage. For example, we looked at the streptolysin O, which is the bacterial toxin. So when we get infected with the bacteria, actually this type of membrane damage really happens in our body. And also we treated the cell with the silica. So silica is like glass, tiny, sharp glass. Therefore, it's a real physical damage. And also we looked at the laser damage. We put the cell under the microscope and hit the very edge with a very strong laser and observed whether the cell undergoes senescence or not. And all these like chemical, biological, and physical and laser optical damage can induce cellular senescence, and that can be suppressed by the membrane repair factor, escort protein. And we also found that that is mediated by the different pathway than the traditional like senescent cell, like DNA damage repair pathway. So we see calcium and P53 dependent regulatory pathways are involved in it. So these results suggest that the plasma membrane damage dependent senescence is a novel subtype of senescence. And we also found, so for the sake of time, I skipped the detailed data, but we also observed that what will happen after the calcium influx into the cell. And what we found is high concentration of calcium ion getting into the cell, and that is taken up by the ER, endoplasmic reticulum, and then that is delivered to the mitochondria via the membrane contact. And actually, mitochondria serves as a reservoir of the extra calcium ion. And that will allow cell to survive. However, if the calcium ion go beyond the limit of the mitochondrial calcium capacity, the mitochondria induce the mitochondrial ROS production, and that would trigger cellular senescence. Therefore, this mechanism is like a double-edged sword. That will allow cell to survive, but at the same time, that will induce cellular senescence. So far, we actually found 3 layers of calcium-dependent mechanism during the process of plasma membrane damage-dependent senescence. So for the cells to undergo senescence, first cells need to survive after membrane damage. Therefore, calcium-dependent membrane repair mechanism is the first layer. And we also found the calcium P53 dependent, the non-canonical senescence pathway. And I also mentioned the mitochondrial calcium overload as the third layer of senescence mechanism. So, all right, now we found novel subtype of senescent cells, which is different from traditional DNA damage or telomere shortening dependent senescence, which is great. But the biggest question is whether that is simply an artifact, can only be seen in a cultured cell setting, or those cells truly exist in human body in an age-related pathology context, for example. And we wanted to compare the gene expression profile of our senescent cell and human body senescent cells. And we did a literature search. Actually, there are so many different locations, so we can see that senescent cell accumulation. And we decided to focus on the wound healing process because, you can imagine like wounding is like a real mechanical stress. Therefore, we hypothesized that mechanical stress can induce plasma membrane damage, and that will induce cellular senescent cell accumulation in human body. To test that, we compared our senescent cell gene expression profile, like transcriptome, and we took the data sets from the publicly available paper, the human body senescent cell near the wounded site. And the human body wound site is like this pink, and our senescent cell is like this green. So actually, if we look at each gene, so the gene is not 100% overlapping. But if we look one layer up, like a cellular process or GO term, they are significantly overlapping. And that overlap is much more significant compared with the control, so DNA damage dependent senescence cells. So we tried like two different analysis method, and the tendency was the same. Always our senescent cell overlap more with the human body wound healing senescent cells. So this is just association, so we cannot conclude any like cause-consequence thing, but one possible explanation is, so our senescent cell, so plasma membrane damage could be the cause of senescent cell induction in human body near cutaneous wound. And actually this was striking because at this moment, no one in this world knows what is a real cause of senescent cell induction in human body. So we know telomere shortening induces senescence, DNA damage induces senescence, mitochondrial dysfunction, many stress induces senescence. However, what's really going on in the body is still like remain open question. And these results raise a possibility that at least in the context of mechanical stress, maybe plasma membrane damage could be the real cause of cellular aging in our body. So very talented student and a former student and currently a postdoc, Jan, decided to perform the biomarker identification because we wanted to ultimately identify our cell in human body in a patient sample. Therefore, we wanted to know the protein that is highly upregulated in human body, in our senescent cell, compared with like young cell, and also compared with the typical other type of senescent cell, like DNA damage dependent senescent cell. And we compared the protein expression profile and identified the protein that is highly and specifically upregulated in our plasma membrane damage dependent senescent cell. And Jan found the quite significant and striking upregulation of wound healing related proteins, specifically in a membrane damage dependent senescent cell, but not in the DNA damage dependent or senescent cell or the young cell. So this strongly supports our working hypothesis that the cause of senescent cell accumulation near the wound in human body could be the membrane damage and mechanical stress. And Jan also performed the bioinformatic analysis and he got a really exciting result. So the protein expression profile of our senescent cell, new senescent cell, are highly associated with various age-related pathologies, like osteoarthritis, joint pain. When you get older, you get a joint pain. That is highly associated with our senescence, and also hypertension or atherosclerosis. So these are also, blood vessel-related, age-related pathologies that are highly associated with mechanical stresses. Therefore, these are quite encouraging to us, because we hypothesize that maybe our senescent cells may explain the cause of those pathologies in our human body in an aging context. So, we found a novel subtype of senescent cell, and that can potentially contribute to the various cellular pathological and physiological processes, like beneficial function, like wound healing, or deleterious, detrimental functions, like joint pain or atherosclerosis. Seems like senescent cell has both good and bad functions. So actually, back in those days, so people thought like senescent cells in our bodies are all bad, so we should just purely eliminate it. So people are trying to develop the senolytics, that drug, eliminating the senescent cell. But looking at that, it's very clear that senescent cell should be divided into multiple subtypes. Senescent cell should be really context dependent. So you can imagine the term senescent cell is like a cancer cell. When you say cancer cell, it's an umbrella term that includes so many different gene expression profile based on its background or stresses or developmental context or whatever. Therefore, senescent cell is also the same. Depending on the cell type or stress type, the gene expression profile should be different. Therefore, each senescent cell should be targeted differently, ultimately. So, another striking result from Yan is that he found that he identified the marker protein for our senescent cell. And when he looked into the database, he found that upon mice aging, our marker proteins are indeed show that kind of upward trend, at least in some tissues and origins. So this is, again, there's no cause or consequence discussion we can make, but one possible explanation is that our senescent cell might be increased in at least some tissue context. So here is our working hypothesis. So there's a plasma membrane damage and cell undergoes senescence. And ultimately, that will contribute to organismal aging in some pathologies. And that can be suppressed by the upregulation of plasma membrane repair process. And we also found that actually upon mice aging, we see the decline of the membrane repair factor expression as well. Therefore, not just plasma membrane damage is increased, maybe the membrane repair factor is downregulated in an aged organism. So that might be a potential feedback loop that could be the answer for how our cells age and how we age in the context of mechanical stress. All right, so our next chapter, so we previously worked mostly on yeast and human cells. But now we are trying to leave our comfort zone, and we are trying to start something very new, which is like organoid and microfluidics research, and also the drug screening. So organoid, as you may know well, is mini in vitro organ, so organ on chip. Therefore, I would like to investigate the phenomena we are looking at in a culture setting or yeast setting can also be observed in an organoid setting. And then we are trying to identify the small molecule that can slow down the senescence processes. And hopefully I will like to ultimately contribute to healthy aging and longevity. And drugs identification, actually we are already having some preliminary results. And Camilla, really talented PhD student, and also in collaboration with Yan, we are focusing on the substances purified from the Okinawan food-derived molecules. So as you may all know, like Okinawa is known as a blue zone, well, previously. Therefore, this is ideal island to study healthy aging. And we are focusing on some of those food-derived products, and Camilla identified several that will selectively either suppress or eliminate senescent cells. And hopefully, some of them could potentially be socially implemented in the future. And also, we are focusing on some substances that is highly expressed in naked mole rat as well. So I would like to understand the secret of healthy longevity of naked mole rat as well. All right, so, and I stop here, and I would like to thank actually my mentors, Professor Tadashi Yamamoto and Professor Yokoyazaki Sugiyama. So they are dual mentors. I really appreciate it, and collaborators, and funding, and the lab members, and actually all the lab members in the corner unit history. And also I would like to specifically mention that my RUA, Hitomi-san, was extremely supportive and one of the most efficient supporter in my lab. And also, last but not least, I would like to thank Yosuke. So he collaborated with me both in the lab and also at home. And without his support, I couldn't have survived. So thank you very much for all your attention, and I'm happy to take any questions.